Hello YouTube, Joseph here with Prep for Step, back with another lecture about major histocompatibility complexes. In the first video, we already talked about MHC type 1 and the general structure and the general function of MHCs. Here we're going to continue with the MHC type 2, okay? So major histocompatibility complex type 2, what is it? First off, it's a protein. It's going to be on a cell surface. It's synthesized in certain cells. The only cells that are going to synthesize this type of MHC and are going to present them on their surface are going to be APCs, or antigen presenting cells. Antigen presenting cells, so this is the general idea. Antigen presenting cells are phagocytic cells, such as macrophages, histiocytes, right? Dendritic cells. These type of cells initially encounter foreign antigens, pollens, microbes from the outside world, okay? Something that's already broken the mucosal or epithelial barrier and now is inside our body, okay? These cells will initially, as they're in the tissues, they will initially encounter these antigens. They will phagocyte these antigens. They will eat them, bring them inside. They will digest them into small little particles, and then they will present these particles on top of their cell surface on the MHC type 2 so you can elicit an immune response from our immune system. That's the whole story, and that's what we're going to go through and we're going to talk about, okay? So we talked about the different types of APCs. This MHC type 2 itself, the structure is made up of two different chains, an alpha chain and a beta chain, okay? This will be our alpha portion of it, and this will be our beta portion of it, or the beta chain, okay? They bind, this MHC type 2 will bind to CD4 cells, or CD4 positive. Again, CD is cluster of differentiation 4. Okay, this is a cell surface marker for T helper cells. Okay, it is also located on naive T cells, so T cells that have yet to be differentiated into either T helper cells or, for instance, cytotoxic T cells. So it could also bind to a naive T cell, but in most cases, most reference books, they suggest that it binds to a T helper cell. Okay, so MHC type 2 binds to a CD4 or a T helper cell. This particular type, MHC type 2, uses the exogenous, or better yet, the better name for it is the endosomal pathway of MHC loading. Okay, so what does that mean? And we're going to talk about that in a second. Okay, so in this section, we're going to talk about the exogenous or the endosomal pathway of MHC loading. Obviously, it's telling you there's going to be an endosome involved. And we're still talking about type 2. Okay, this is particularly only used for type 2, not for type 1. In this case, we're using an invariant chain. If you remember from type 1, we were using a tap complex. Here, in the endosomal pathway of type 2, we're going to use an invariant chain. So as we go through the story, you'll understand everything will fall into place and where this invariant chain comes in. Okay, so this antigen presenting cell, this is not any cell, this is an antigen presenting cell or a phagocytic cell such as a macrophage, is roaming around inside our tissues, and suddenly it encounters a foreign antigen. This foreign antigen is then going to be engulfed. In most cases, once it's broke down, it could be actually exocytosed. But in some cases, when it's necessary to elicit an adaptive immune response, we need to somehow break this down into small little particles, break this foreign antigen into small little particles, and present it on an MHC type 2. Now, just like an MHC type 1, the MHC type 2 is still getting synthesized. Both chains, the alpha and the beta chain, are both getting synthesized in the rough ER. But this peptide binding groove here, we're trying to prevent that from our normal peptides. Remember, in MHC type 1, we wanted the normal peptides to bind to this groove. In MHC type 2, we want these foreign guys to attach to this particular section. So what we will use, at the same time as we're synthesizing an alpha and a beta subunit of MHC type 2, we're also going to synthesize an invariant chain. This invariant chain is going to sit on this peptide binding groove so it stops any regular cell peptide from binding to it. So we can specifically have our foreign peptides bind to it so we can elicit an adaptive immune response, okay? So as it's leaving the rough ER into the Golgi complex and all that, it's designated for the cell surface, it's going to come into an endosome, okay? It's going to be covered by a membrane, a vacuole, with the invariant chain, as you can see here, binding to the peptide groove, okay? So once that happens, now these two guys are going to fuse. Okay, so with the foreign antigens and the MHC type 2 in that vacuole, they're going to fuse together. The acidity inside this vacuole will cause this invariant chain to get removed from that groove. The acidity is what actually removes it. And then the antigen particles, the small little antigen particles, whether peptide or not, it doesn't really matter, but these particles will actually bind here, and this will fuse with the membrane in the next step, and it will be presented on the antigen presenting cell. Over here in this particular section, we're trying to present this antigen to our immune system to get a response. In the MHC type 1, we were presenting self proteins to try to stop it from killing us and just telling, we're trying to tell the T cell that, we're self, so do not attack us. Here, we're trying to tell the T helper cell that this is a foreign antigen, so start the adaptive immune process. So if the T helper cell, which is CD4 positive, remember it's CD4 positive, and the TCR, let's say this is the TCR, the T cell receptor, will bind to this groove with the help of the CD4, and this is our CD4 molecule, will help this, and based on this idiotype, based on this 3 dimensions here, and based on what type of antigen is presented, the T helper cell would then decide whether it's going to need a humoral response with antibodies, such if it was a bacteria, or if it's going to need, let's say, a viral, a cytotoxic T cell, or a cell-mediated response. Okay, so that's the invariant chain pathway, or the endosomal pathway, and that concludes the discussion of MHC type 2. I hope this was helpful. Please share, like, subscribe. You can check us out on Facebook as well. Thank you, and have a great day.